Good morning. Let's go ahead and get started. Thank you all for coming today. Some of you noticed that we opened up the cookie bar early. If any of you want to come grab something as we're getting started, feel free to do that. The goal will be to not take any of that out of this room from our end. So really appreciate you coming. Excited to have Dr. Stephen Greenberg with us today. How many of you were at his talk last night, just out of curiosity? Okay, just a few. So coming back for more, we had a great talk last night. Today he's going to talk about taking the shortcut, biomarkers for cerebral small vessel disease. Let me give a brief introduction to Dr. Greenberg. Dr. Greenberg is a professor of neurology at Harvard Medical School, vice chair of neurology for the Faculty Development and Promotions, and holds the John J. Conway Endowed Chair in Neurology at Massachusetts General Hospital. He is recipient of the 2017 American Heart Association William M. Feinberg Award for Excellence in Clinical Stroke, and has served in many national and international leadership roles in the fields of stroke and neurology, including the principal investigator for the NINDS Mark VCID Biomarkers Consortium Coordinating Center, which he talked a little bit about last night. Very interesting research. President of the International CAA Association, chair of the NIH Acute Neurologic Injury and Epilepsy Study Section, co-chair of the NINDS Alzheimer's Disease-Related Dementia Summit Subcommittee on Vascular Cognitive Impairment, and chair of the American Heart Association International Stroke Conference. Dr. Greenberg has authored over 200 peer-reviewed research articles and over 75 chapters, reviews, and editorials in the areas of hemorrhagic stroke and small vessel brain disease. He received his undergraduate degree in biochemistry from Harvard University and MD and PhD degrees from the Columbia University under the graduate research training of Dr. James Swartz. He performed internship at Pennsylvania Hospital, neurology residency at MGH, and postdoctoral fellowship at Brigham and Women's Hospital under the training of Dr. Kenneth Kosick. Please join me in giving Dr. Greenberg a warm welcome this morning.
Well, thanks very much. I'll semi-apologize people who were at last night's talk. There'll be a little bit of overlap and really a lot of the same themes, but I get a little more science-y this morning, and one thing I'll overlap from last night is my gratitude of being invited to speak at this conference, and just how really impressed I am with the institution as a whole, but in particular the seriousness and thoughtfulness and commitment of the gerontology program and of the faculty and students who I've met during my stay here. So really, I'm going to leave here with a lot of excitement and positive feeling about the time. I mentioned last night this work supported by the National Institutes of Health. By way of disclosures, I will be talking a little bit at the end of my talk about a clinical trial in amyloid angiopathy, and so I mentioned as disclosures that I am a consultant for some other trials in the immunotherapy approach to Alzheimer's disease. And I'll barely touch on that, but it's a really interesting area of using antibodies to try to prevent or treat Alzheimer's disease. And the trial I'll talk about is one where I wasn't paid directly as a consultant, but my institution had a research agreement with Pfizer, the sponsor of the trial. And I showed something like this last night. This is a slightly different list, and of the people I've worked with, both on the bottom, the people from Pfizer I worked with on the clinical trial, but and at the top, this Mark VCID consortium I'll talk about, and other colleagues from my institution. And I really am repeating what I said last night, but every time I speak, I make the point that patient-based research is a team sport. And so for anybody who's interested in lab research, I think it's sort of possible to go it on your own, order up all the fruit flies yourself and have your own lab. But anyone who does research on patients does it collaboratively because it's hard to do. Each patient you enroll in a study is a real personal effort, and the way this happens is not by one person, but by a community of researchers, both within an institution and across institutions. So I really encourage you to think socially about the building a network, as hopefully some of you will build careers in patient-based investigation. I showed this slide last night. I won't go over it again, but this is just sort of the motivating point. This is the numbers from the Alzheimer's Association in terms of what the amount of dementia will look like. And I'll talk a little bit this sort of what's Alzheimer's disease, what's dementia? Well, dementia is the problem. I mean, that's age-related memory loss and cognitive loss, and it is the public health problem of our time. It's the term Alzheimer's disease often gets used synonymously, but that's clearly not the case. And as I'll talk about and was more of a topic last night, that we now understand that dementia is usually more than one thing happening at the same time. Alzheimer's is a big part of the picture. I'll be talking about other parts of that picture. And in response to the public health threat that we have in 2010, the administration proposed, Congress passed the NAPA, the National Alzheimer's Project Act. And for people like me who study other conditions that contribute to dementia, I put this act has a whole bunch of text, but this is the part that we all paid attention to, was the inclusion of what's called the Alzheimer's disease-related dementias. Gets everything in Washington gets an acronym, gets an abbreviation as the ADRDs, and these are the ones identified as ADRDs: frontotemporal dementia, Lewy body dementia, mixed dementia, and vascular dementia. And I was at this conference Jeremy mentioned in the introduction, this ADRD summit, and some of the people, scientists, are kind of an ornery, argumentative lot. And so somebody raised their hand and said, "Why do you keep calling us the ADRDs? I mean, we're not really exactly Alzheimer's related. Each one has our own disease. Why do you keep sort of saying we've got to, we have this name Alzheimer's related?" And the NIH program officer said, "Well, there's a very good scientific reason for that. That's how you got the money." And meaning that by being sort of brought in under this Alzheimer umbrella, that's was how some of this Alzheimer money was spread around. And you'll tell from my talk, I think it's very appropriate, because again, this idea that dementia is more than one thing, and often overlapping dementias. And I showed some of this last night in my talk, that this is work from one of many studies that have really tackled the problem. This is the Religious Order Study and Rush Memory and Aging Project that's run out of Rush in Chicago, Rush Medical Center. David Bennett and Julie Schneider, the key investigators there. And what you're looking at in the upper left-hand corner, so these are people who were tested during life and then agreed to donate their brains for autopsy, for pathologic analysis. And all those lines on the ground, all those single little lines, there are 236 of them, represent different combinations of pathologies. And laid out in this table are how many out of 1,079, so about 1,000 brains, how many of them had each of these different pathologies. So AD, Alzheimer's disease, is at the top of the list. And this isn't just, this is the way they define in each case as having enough to be related to your cognitive impairment. And then they did multivariable modeling, and if present, this is the amount of your cognitive decline that you could attribute to that pathology. So it's kind of a complicated table, and the way I think of it, it's sort of, if you multiply these two numbers together, you get an idea of how big the impact of each of these pathologies. So Alzheimer's disease definitely wins by this analysis, but these other things, and just, I put a box around the ones that are vascular, that relate to blood vessel conditions. So gross infarcts, and I apologize when I go medical in the language here, it's, but infarct meaning a stroke, an area of damage to the brain caused by blood vessel conditions. Amyloid angiopathy, which I'll talk about at good length. Atherosclerosis, so that's hardening of the big arteries, and arteriolosclerosis, which is hardening of the small arteries. And so it's actually quite a big part of the problem. So, so the take-home lesson here is that the blood vessel contribution to cognitive impairment is really quite substantial, and probably behind the Alzheimer's contribution, but again, if you sort of work the numbers out, not way behind. I mean, it's really a big part of the picture. So we had this summit, and there's sort of a drumbeat. Each of the groups kind of peeled off for each of those things: frontotemporal dementia, Lewy body dementia, mixed dementia, and vascular dementia. And we issued recommendations, and we didn't really get together to coordinate our recommendations, but each one had this biomarker theme that was just kept coming up, and that's what I'll talk about over the next few slides. And really the question is, sort of what's the magic about biomarkers? Why are we all so obsessed with it? And what it really comes down to is that I don't know how many people in the room would sort of identify themselves as being interested in studying the brain, but how many people would sort of identify themselves? So yeah, so a few, and well, y'all picked a tough organ because it's behind this wall, not so much the skull, but behind this wall called the blood-brain barrier, which everyone's familiar with, and it's a very tough organ to study. We're all jealous of people who study the blood because it's right there. You can take a blood sample and study it, and so biomarkers is really the equalizer, the way to get clues in living patients. I mean, we can do a lot of things at autopsy and looking at the brain, and I'll show some pictures like that. But of course, the people we want to study and the only ones we can help are people who are alive, and especially when it comes to clinical trials, which I'll talk about. So anyway, that's the introduction to the three topics I'll talk about over the remainder of my talk. So sort of general biomarker considerations with regard to this NIH consortium, MARC-VCID. I'll then talk about applying some of these ideas to amyloid angiopathy, and then I'll close in the last few slides with a clinical trial where we used a biomarker, and the results weren't exactly the ones we wanted, spoiler alert, but they're still sort of illustrative of potential themes in the use of biomarkers for clinical trials. So, well, I did what any self-respecting person would do when I started giving talks about biomarkers. I Googled biomarker and came up with a definition, and this actually isn't such a bad, it gives you the pronunciation and the speech and all that stuff, but it actually sort of covers the main features. I mean, that it has to be something that's measurable and has to indicate something, and that's actually not such a bad place to start. So the measurable part, as I say, is the tough thing about the brain is getting access, and I'll talk a lot about MRI, and that's really been, in a way, the great equalizer because MRI has just been this remarkably flexible tool for being able to look at different aspects of brain anatomy and brain function. But whatever your biomarker is, it has to be something accessible that you're able to measure, has to be reproducible across time points and sites. And we have a lot of problems with, one person will measure something at their site. I mentioned that this is all done in collaboration and it's a team sport. And if you can't get on the same page with somebody at a different site, it's not helpful. And it has to actually, measure the thing you're trying to measure. And then it's indicative of some phenomenon. And I'll talk a little bit about this. What do you do with these biomarkers? And this is the list that we've come up with in our consortium that I'll talk about in a moment. So the first three things on the list are to work out the kinds of patients you might enroll in a trial. So having a risk factor for a disease, or diagnosing you and saying, oh, you look more vascular or less vascular, and prognosis or stratification, or saying what your likely future course is going to be. So those would be sort of features of an individual. And then if you do some kind of an intervention, what are the ways that you measure response? So mechanism of action, I should say target engagement, is if you do a treatment, is it just even doing the thing the simple thing that you're asking it to do? If you give a drug, is it even getting into the brain, or reducing inhibiting the enzyme that you mean to inhibit? Mechanism of action is sort of a broader concept of, are you changing a pathway or changing a larger part of the disease pathogenesis? Disease progression is, now you're getting at, are you really slowing the disease? And that last thing that I have in parenthesis, surrogate marker, that's sort of the highest bar. That's when you're saying your biomarker is so good that if I do a clinical trial and I'm able to improve the biomarker, then we're done. I've shown that my drug works, and that's a really high bar that isn't achieved by very many biomarkers. For the most part, when you try to get your treatment approved and into general use, it's the biomarker is more the hint that you can then go on and do a big trial rather than the surrogate endpoint. So this is the consortium we put together. I'll just talk over a few slides. It's not this is a big part of my life, but we don't have much to report yet because we're still in early days. This is this MARC VCID. VCID stands for the Vascular Contribution of Cognitive Impairment and Dementia. It really should be VCCID, but trying to shorten it up a little bit, so it's VCID. And the goal is this is really to develop what we hope will be a suite of biomarkers that can be used in clinical trials of small vessel disease. Here we are, we're somebody pointed out we're pretty coastal, not completely, but for better, for worse, but across 7 strong centers. Some of those centers themselves have multiple members. We have an external advisory committee, and I'll thank here Rod Corvo, who's been the program officer at one of the National Institutes of Health, who's really championed this model. Here we are at we had a face-to-face meeting in Los Angeles coinciding with the International Stroke Conference this past year, and we're set up to have kind of a 2-phase effort. And again, this isn't so exciting, but for 2 years, it's what I call the let 100 flowers bloom phase, where sites are refining their candidate biomarkers. And then over the last 3 years that we're just going to enter soon, and we'll prove that these markers can be done across sites and that you can get reliable results. And this is my editorial comment at the bottom, that the hope is that what's year 6 of a 5-year plan? I mean, my hope is that year 6 of the plan will be to actually start to apply these biomarkers to clinical trials. So right now what's happening, we're in that transition period, and the sites are each proposing their biomarker, and I've shown that's supposed to be a smoke-filled room, because what's going to happen is all these sites are going to propose, here are the biomarkers that we think are ready to go, and that's my friend Rod Corvo at NINDS, the one of the NIH agencies that's funding this work, and Walter Korschetz, who's the director of the agency. And what's going to come out of all this is NIH is going to publish a list. They're going to have sort of tier 1, tier 2, tier 3, and that's going to be our marching orders, and really sort of the marching orders for the vascular cognitive impairment community. Those are going to be the biomarkers that they're going to say, we want you to go and validate them. So what are the criteria that are going to go into that decision? Well, again, this is a little bit of sort of nuts and bolts, and we don't, I don't have the payoff here to tell you what these markers will be, but it's got to have a biological link to the thing you're trying to study. It has to be useful for a trial, and it has to be ready for cross-site. You have to show that you can measure the same thing across multiple sites. And right now, the feeling is that there aren't any biomarkers that have met those criteria yet. So which ones are already in the bank? Well, really none of them. So just to walk through briefly what each of those things means. So biological link means is a real issue, because we're very good at saying that something is associated with vascular disease, but we want better than associated. We want to say it's really a key player that, and this is a tricky issue to understand, but it's an important one for anyone interested in research, is you show that some marker is increased at a, for people who are used to writing these papers, p less than 0.05, that's sort of the cutoff to show that you've got a significant relationship. But that's not good enough. Really what you want to show is that the marker is catching one of the things that's not just a bystander, but part of the real pathogenic process. And I'll talk about what I mean by that a little bit more. I'll flesh it out a little bit more with regard to amyloid angiopathy. Second thing is usefulness for a trial, and it's those same categories I showed before. I won't go over them again. And it may be that different markers will apply. There may be one marker for very early or pre-symptomatic disease, another marker for later disease. And then the third thing is to be able to show that you can validate it across sites, and there's this kind of technical validity, just can you get the same thing reproduced across sites. So anyway, we're at a stage where we're ready to take the dive into proving biomarkers are meaningful And so I hope if you're kind enough to invite me back in a few years, I'll be able to sort of show the list of biomarkers that we've been able to validate. Well, let me try to flesh out. Right now, everything I said is sort of up in the air. This is what we're trying to do. So let me flesh it out a little bit more with regard to the disease that my group has studied. And I talked about some last night, this thing, cerebral amyloid angiopathy. So it's one of the 2 common small vessel diseases. I'll walk people through it a little bit. So what you're looking at in the upper left-hand corner there, so these are all we call histopathologic sections. So it's the brain that's been sliced into ultra-thin sections and then stained and then looked at under a microscope. And so what you're looking at here is it's a blood vessel, but it's a blood vessel in what we'd call longitudinal sections. So the blood is flowing off the bottom of the slide here. That's what we call the lumen of the blood vessel. This is the wall of the blood vessel. This is the outside up at the top. And you wouldn't necessarily know it, but almost everything you're looking at here is normal with the exception of these little circles here. And what these are, that is the amyloid peptide, the culprit in amyloid angiopathy that has deposited right on the outside of the layer of the vessel wall that contains the smooth muscle cells, the cells that account for the properties of a vessel, be able to constrict or to dilate. And the person who taught me this is a Swiss neuropathologist named Jean-Paul van Zettel, and he would call those holes in those donuts the tombstone of the smooth muscle cell, because our understanding is that this amyloid engulfs around those smooth muscle cells, and then they die right there, sort of in front of your eyes. And I always bring this up because in Alzheimer's disease, it's not obvious necessarily where the amyloid deposits versus where you have death of brain cells, of neurons. But in amyloid angiopathy, you can see exactly where the cells used to be. And as you get over to this figure on the right, now essentially the full thickness of that smooth muscle cell layer has now been replaced by amyloid. And this is this classic staining property that it turns green under with a particular stains, this thing called Congo red, under polarized light. And that reflects, for people who are sort of biochemically inclined, that reflects the beta-pleated sheet structure of the amyloid deposits. So far you might say, all right, this isn't so bad. You've lost those contractile abilities, but at least there aren't any holes in the vessel wall. But you get more advanced stages where there's actually that amyloid actually causes breaks in the blood vessel wall, allows junk to begin to leak in and eventually to open up and bleed. So actually bleeding is the way we often recognize this amyloid angiopathy. And for people who are familiar with the Alzheimer's world, this is where amyloid angiopathy fits into Alzheimer's disease. And so this A beta in the middle here, this is that amyloid peptide that's produced from a larger precursor protein, the beta amyloid precursor protein. It can deposit in the brain tissue as plaques, or can go through one of these clearance pathways, can be degraded by proteases, can be brought out across the blood-brain barrier in going in to out. Or the pathway that we study the most is this interstitial fluid drainage pathway, where there's actually an old literature that it had been shown years before that if you just inject any kind of dye, India ink, anything into the brain, it ends up lining along the blood vessel walls. And we think that's how it gets access to the blood vessel walls to create amyloid angiopathy. And though I mentioned bleeding, I'll just mention here, this is again work from that same religious order study, memory and aging project, showing that having amyloid angiopathy not only causes bleeding, as I'll talk about, but also is this contributor to cognitive impairment. I kind of mentioned it earlier as well, that either in sort of a cross-section when you test people, or if you follow their trajectory over time, that the more of this you have, the greater cognitive impairment you have over time. So the way we recognize this typically as neurologists is by the bleeding. And the reason that we're able to focus on bleeding a lot, I always say to people, the M in MRI stands for magnetic, and the blood has iron in it. And so what that means is that not only can you see big areas of bleeding, this would be considered a hemorrhagic stroke, a bleeding stroke, but you also see these micro bleeds, as shown in the arrows, or this thing we call superficial siderosis, where you get blood following along in the sulci of the brain, the folds in the brain. And these would be really hard to detect with any technique except for MRI, but because MRI is so sensitive to iron deposits, and actually there's what's called a blooming effect. It actually looks bigger on MRI than it looks than the actual thing producing the signal. We're able to detect it. So, and this is the basis for how we diagnose amyloid angiopathy during life. This is really a first step that is key to, you can't study it in life if you can't diagnose it in life. And the criteria shown in blue are, that are the Boston criteria for probable amyloid angiopathy. And I won't get too much into this, but you need multiple bleeding lesions that are all out near the surface of the brain in the typical spots for amyloid angiopathy. And that is now in common use both for research and for just clinical diagnosis of the disease. So, so now let's talk about, so what are the biomarkers for amyloid angiopathy? Well, I mean, bleeding is really number one. And I'm using this term, actually I borrowed the term from a colleague in Utrecht, Geert-Jan Biesel's markers of brain state. So what we're really marking here is some kind of injury that's taken place to the brain that we can look at after the fact. So, so that bleeding is the starting point, but it's not the finishing point, that there are all these other non-bleeding markers, and this gets back to the part I mentioned before about you're not only causing breakage in the walls of the blood vessels, but potentially disrupting the ability of the blood vessel to do its job properly. And so these other lesions, and I won't go over them in great detail, represent more the mucking up of the normal vessel function, and I'll focus on a few of them. But it's this. I'm repeating this line that I had earlier in the talk, which is that the real trick is to decide which of these are the important ones, which are actually contributing, especially to the cognitive impairment. And the one I'll start with, and I talked a little bit about last night, are these microinfarcts. This is really an interesting area. And so these are, so the term infarct means, death of cells. This isn't a bleeding event. This is from a loss of blood flow. And it's clear that these things are so truly micro, so 200 microns, so smaller than you can see with your eye. You would really, and actually that smaller than you can see with your eye thing is a big problem, because a neuropathologist will look at the brain, and if they can't see anything grossly with their eye, then all they can do is cut some sections and hope they'll find something. But you can't cut thin sections of the whole brain, so you're only sampling a few places in it. That's actually a big issue, and I love to show this because the author of this study, Brandon Westover, is a proud BYU graduate from 1999 and is a rising star in actually in the epilepsy field, but loves any mathematical problem I can throw at him. So I asked him the question of, well, gosh, since you're only able to see microinfarcts in those few sections that you look at, how many do there have to be in the whole brain for you to see anything? So we kind of worked out numbers, and we published this paper together a few years ago showing that if you only see one or two in the sections that you cut, it means that there are hundreds or thousands in the brain as a whole. And I heard it expressed by a pathologist. I put this quote up. Anybody live in New York before? So if you see one cockroach in your kitchen, it means there are thousands in your walls. And when I heard this pathologist say this, I couldn't sleep that night thinking about it. But the point is that these are really extraordinarily numerous lesions in the brain, and the real problem is we're just not good at seeing them, and that's what makes it a limited biomarker. We have a couple ways that we take a shot at it. One is my colleagues in the Netherlands, Geert-Jan Biesel's and Susan van Velu, who's now with us in Boston, use very powerful MRIs, these 7 Tesla magnet MRIs, to see at least a subset of the bigger microinfarcts. It's always a great oxymoron. It's like a jumbo shrimp. But you can see at least a small tip of the iceberg. And the other way is that for people, this is a commonly used tool in the stroke field, something called diffusion weighted imaging will show you very clearly a new stroke, but only a new one, only within a week or two of when it happens. You see these little white dots on a different kind of MRI. I mentioned MRI is a very flexible tool and is able to detect these, but we know we're just seeing a fraction of them. I'll mention here that there's one thing we can do in research that is quite hard to do in real practice, which is when we see one of these little acute small strokes, or one that just happened, because our patients are involved in research studies, we're able to do sort of a before and after comparison. So we can look at the MRI before they had that small stroke and then after. And we can sometimes make out a tiny hole, not always where it happened, but it's one you'd never be able to see unless you knew right where to look. Again, we have the advantage of looking there. And we used another technique that I mentioned last night, this thing called diffusion tensor imaging, to look at how it affects the microscopic structure of the brain in that region. And I won't be able to go into it in great detail, but what we see is that it disrupts the structure. It causes increased diffusivity, which means less organization of the tissue, or decreased anisotropy, again, a marker of less organization of the tissue. So even though it's tiny, it has real effects on the way the brain wires up. And that sort of leads to another one of these markers, which is altered connectivity. And so this is now again, I mentioned last night, we use different techniques, including functional MRI, but here this is structural MRI, diffusion tensor imaging, to look at the way one part of the brain connects to another. And this is a technique that's well worked out. This is work that Yael Reimer did as a postdoctoral fellow in our group, dividing the brain into these nodes, these small parcels of the brain, and then looking at the connections between them and coming up with a measure called global efficiency, which is a global measure of not only where there's loss of structure, but also how central that pathway is. And I showed this last night. This is really the best marker in our hands of cognitive impairment, that this global efficiency measure correlates quite well with particularly executive function, processing speed, and it also progresses over time. So the concept here that I want to emphasize is that when you look at global connectivity, you're kind of saying, I can't count all the individual lesions. I can't find all these thousands of microinfarcts. So instead, I'm going to aim for a property that kind of subsumes the cumulative effects of a large number of distributed lesions and produces an overall signal. And that may be the way we need to go. So this is the kind of biomarker question we have, which is, do you want to sort of go lesion by lesion, or do you want to aim for a less specific but a more comprehensive measure? And there is no one right answer to it. I won't go through this, and I think I'm going to cut short a little bit in the interest of time, because we're going to run short quickly. But this is another diffusion tensor-based approach, graphing histograms of this diffusivity. I think what I'll do is cover one more topic and then maybe stop and open for questions, because I'm conscious of running long. So, what I've shown so far are what I call markers of brain state, or the term that I'm using. But you also know about markers of brain processes. So let's say, all right, instead of looking at kind of the structure of the brain, I want to look at some of the processes that I think are important in vascular cognitive impairment. And for amyloid angiopathy, there are really two processes. One is amyloid deposition, which I won't talk about it, but it's a really interesting tool to be able to measure amyloid using PET scans with ligands that specifically stick to amyloid. The other one that I did mention last night is vascular reactivity, the measuring the ability of blood vessels to dilate when there's requirement for increased flow and constrict when there's not. And I showed this picture last night. So what you're looking at here, functional MRI measures the increase in blood flow to an active area of the brain. So the gray part of the graph is the increase in blood flow to visual parts of the brain, the occipital cortex, when our patients are in the MRI scanner and looking at a screen with a flashing checkerboard. And the salmon-colored curve is the increase in blood flow, the normal increase in blood flow for elderly controls. The blue color is that increase in blood flow for people with amyloid angiopathy. And so you see, it does happen. I mean, it exists, but it's both smaller and slower. And these are the numbers we measured at the bottom, that there's both a lower amplitude peak and also a longer time to peak, a longer time to return to baseline. Our colleague Eric Smith in Calgary found essentially the same results. And you might worry, oh, well, wait a second, if you get some Alzheimer's changes too, maybe this is not so much a difference in the blood vessels, but a difference in the way the neurons are working. And we more recently used the fact that there are a small number of families that inherit essentially pure Alzheimer's disease. And this is this Dutch type amyloid angiopathy, or that HCHWAD is the Dutch hereditary form of amyloid angiopathy that's basically an autosomal dominant pure form. This happens instead of people in their 70s, happens people in their 40s and 50s. And what you see here again is actually even a more robust difference. And it's not so much that the CA patients are doing worse, it's that the controls are doing better, because now the controls are 40 and 50-year-old people rather than 70 and 80-year-old people. And we saw that you could see the same differences either in patients with who've already had bleeding strokes, so people who are symptomatic with the disease, but actually even pre-symptomatic people who have the mutation, have some of these changes happening in them, but have never had a stroke, have no cognitive impairment. So it seems to be quite an early step, and this gets at the point that I made earlier, which is that we're looking for early markers, ones that we could potentially intervene on early in the disease. So I'm actually going to stop there. I know I had promised to go further, and I'll be happy to talk more, but I think this is probably a good place to stop to give a sense of the application of biomarkers to a particular small vessel disease. And as you can tell, for all these stories, this is really the beginning, or at most kind of the late beginning of a much longer story, that we're beginning to get at this kind of step-by-step. We almost have an embarrassment of riches. We almost have too many biomarkers, but the question is which ones are the ones that we can use to identify who should be in a trial, and identify whether a trial of a treatment looks promising and enough to advance it to a full-blooded clinical trial. So I think if it's okay, I think I'm going to stop there, and if there's time for questions, I'm happy to take them. But thank you very much for your attention.
We absolutely have time for questions.
Hi, Dr. Greenberg, thank you. What's the difference between coming up with diagnostic criteria for CAA and discovering biomarkers for CAA? In your diagnostic criteria, you're using imaging to make the diagnosis, and the biomarkers are based on imaging as well. Can you tell me, how are those two processes different?
Yeah, so everybody can hear. So the question is, isn't it sort of the same thing to talk about diagnostic criteria and to talk about biomarkers? And yeah, of course, you're right. In that list of uses of biomarkers, diagnosis of subtype, is one of the definite uses. It's of course not the only use, and so you may want to use one kind of marker to identify the condition, and then another one to measure whether it's progressing over time, or to identify a key mechanism that you think is, like vascular reactivity, that you think is key. So, but it's almost a biomarker condition, and you run into danger of circularity. In other words, if you have one biomarker that sort of proves another one, well, can you hang your hat on any of them? And ultimately, the last word is comparison to pathology. So what I mean by that is people talk about the gold standard. What's the one thing that you can hang your hat on and say, yeah, this is definitely amyloid angiopathy? Yeah. And for us, that's involved showing that those microbleeds and siderosis and that stuff correlate with the pathology of having the disease, and then we can begin to use the microbleeds as pick out the patients, and then look to these other biomarkers to say, for instance, with microbleeds, don't particularly tell you who's going to be cognitively impaired. So in other words, I show that global efficiency measure as being the one that really corresponds to cognitive impairment. Microbleed count doesn't. So we have these people walking in, you see these scans with literally hundreds of microbleeds, and you say, oh gosh, this poor person must be ready to keel over, and they're talking to you and telling you what was in the newspaper the day before. There's probably a distinction between the ones we can use to make the diagnosis and the ones that tell you which are the key steps that are actually causing cognitive impairment or predicting future risk of bleeding, or that show whether a drug treatment is working. So I think you're right, but I think it's a bigger picture for biomarker use.
Dr. Greenberg, you were talking about the amyloid beta deposits versus the plaques. So small vessels. Is this an either-or thing, or is this usually if you have them in vessels, you're most likely going to have them actually?
Right, so the question, yeah, so if everybody can hear the question is what's the link between amyloid depositing in the blood vessels versus amyloid depositing in the brain substance itself, in the plaques that are associated with Alzheimer's disease? And the answer is the two processes are clearly linked, but you tend to get one more than the other. So the majority of our amyloid angiopathy patients are not demented, don't have dementia at the time, at least before they have their hemorrhage, and the majority of Alzheimer's patients don't have much by way or any by way of bleeding. So something tends to shunt you down one pathway rather than the other, although they definitely can overlap, and they overlap more than just by chance alone. I mean, that there definitely is a link between them, but it's really a subfield of itself that's mostly been studied in animals. Is, you know, if you remember that sort of complicated slide I showed towards the beginning with amyloid in the center and the different ways that it can go, the factors that tend to say, okay, you're going to deposit in the blood vessels or you're going to deposit in the brain are only partly understood. They're probably genetic factors that tend to push more one way than the other, and probably other things that we still don't understand. One thing that's clear is that amyloid likes to stick to amyloid. So once one process gets going, it tends to snowball. So I think that kind of amplifies the differences that the strongest way to get more amyloid deposits in a place is to have a seed, a nidus of amyloid deposit there, and then it keeps going and going in kind of a domino fashion.
To follow up on that, you said there must be something that kind of tips people one way or the other for the most part. Either it aggregates in the blood vessels or in the brain with some overlap, but not total overlap. Do you think it would be important to try to find out what that something is, or why people tip one way or the other? And how would you go about discovering?
No, we don't care at all. Oh yeah, no, we would love to know that. Yeah, it's I mean, and for sure it's an ongoing question, as I say, and we have made progress. It's not an area where we've made nothing. This I can talk more about if you want. This apolipoprotein E, which is have people heard of it? ApoE, I mean, it's the single strongest genetic risk factor for sporadic Alzheimer's disease, for non-autosomal dominant Alzheimer's disease, and it's also an important risk factor for amyloid angiopathy. But the isoforms are a little bit different. There's this one called the ApoE2 that's actually a risk factor for amyloid angiopathy, but is protective from Alzheimer's disease. So that's the kind of thing that's a clue. And the one I showed this, actually the slide I'm on right now, this Dutch type mutation gives you essentially pure amyloid angiopathy. So there's a point mutation within the amyloid peptide that targets it for vessels rather than for plaques. So that tells you that it can be the structure of the peptide itself that can do it. So it's a great question. A lot of work's gone into it, and we have an outline of an understanding, but not really a full picture. Great. Again, thanks very much for your time.